Welcome to the Antimicrobial Stewardship Project webinar series, created by the Center for Infectious Disease Research and Policy at the University of Minnesota. I am Dr. Marty Peterson, your host today. We are pleased today to have with us Dr. Gail Hansen, who will be our moderator, and Dr. Amy Prudent, who will be our presenter of her webinar titled Dissemination of Antimicrobial Resistance in the Food Chain and the Role of the Environment is the title today. To begin, I'd like to introduce our moderator, Dr. Gail Hansen. She's a public health veterinarian who works primarily as a policy consultant. She has expertise in antimicrobial resistant related topics to food, animals, and the connection between antibiotic use in agriculture and resistant human infections, as well as zoonotic diseases and animal welfare. She has worked nationally and internationally with government agencies, members of food, animal production systems, the pharmaceutical industry, university, non-government organizations, physicians, as well as veterinarians. She has also worked at the Pew Charitable Trust, on Capitol Hill as an epidemiology at state and local levels, and in private veterinary practice. She's particularly interested in the One Health approach to antimicrobial resistance and infectious diseases. And I'd like to also add that she's also been a very important faculty advisor to the Antimicrobial Stewardship Project at CIDRAP. So with that, I'll hand it over to Dr. Hansen, and she can introduce our presenter today.
Thank you. When we talk about One Health, we often tend to focus on the animal or the human side of the antibiotic resistance question. We sort of neglect the environmental health aspects. So I'm really excited to introduce Dr. Amy Prudent, who will talk about antibiotic resistance as it relates to the environment. A little bit about her. Dr. Pruden is the W. Thomas Rice Professor of Civil and Environmental Engineering at Virginia Tech. Her work focuses on bringing a microbial ecological perspective, and she's widely known for her work documenting antibiotic resistance genes as environmental contaminants. Her current research is funded by the National Science Foundation, USDA, Water, Environment, and Reuse Foundation, and the Alfred P. Sloan Foundation. And it focuses on advancing practical means of antibiotic resistance monitoring, mitigation, and risk assessment in all types of water systems. And among other projects, Dr. Pruden is the principal investigator on a USDA project on antimicrobial resistance from farm to fork, which is how I got to know her. She's authored over 125 peer-reviewed articles, and she's an associate editor of Environmental Science and Technology. Dr. Pruden, look forward to your presentation today, and I'll turn it over to you now.
Thank you so much for the introduction, Gail, and it's a real pleasure and honor to be your webinar speaker today. As Gail mentioned, much of what I'll present today is part of a large USDA-sponsored project, and so there are many contributors. This was very interdisciplinary, and I do want to acknowledge some of my co-authors that contributed to some of the material that I will present today. So that includes Catherine Knowlton, Leanne Cromades, Kang Sia, Monica Ponder, Cully Hessian, and Liqing Zhang. And you see also there were several postdocs and students that contributed to this work. And here are the principal investigators across the project as a whole. And also I wanted to acknowledge the contributions of our advisory board, and that includes Gail, and it was very kind of her to suggest that I present today's webinar. So antibiotic resistance, I think we all know this is a huge challenge that we're facing in this century. We've come to rely on antibiotics, not just for human health, but for animal health and food productivity. But we see that it's a double-edged sword, and microbes can develop resistance. These are some reports that are out there that have documented this. So the CDC estimates that about 23,000 people die from deaths that can specifically be attributed to resistant infections. In the EU, it's estimated to be about 25,000 deaths per year, and in India, 58,000 newborns alone die from sepsis because of antibiotic resistance. And so this is a big deal. The O'Neill report coming out of the UK has estimated that by 2050, we'll see deaths from antimicrobial resistance surpassing those of cancer. And the World Health Organization has been a big player in really viewing this as an integrated problem, and they've urged coordinated action in addressing the spread of antimicrobial resistance. I also wanted to mention this United Nations report that came out about a year and a half ago from their Frontiers group that looked at emerging issues of environmental concern. And I thought it was quite interesting that they cited antimicrobial resistance as the #1 emerging challenge of concern for the environment. And this is a figure from that report that I think nicely illustrates what an integrated problem we're dealing with. So we see people swimming in the water, we see livestock, agricultural contributions of antibiotic resistance, industrial inputs, wastewater treatment plants, which is where everything that humans and industry uses ends up when it goes down the drain. And so we see the water and the environment in general as a conduit that could potentially contribute to the spread of resistant organisms to humans. And this is a more simplified cartoon fate diagram that we've been working from in our group that considers some of the broader sources. And so, faded out in the background are routes of transmission of resistant infections that we know about. So people to people, food to people, livestock to people. This has been documented to happen, but we're really interested in understanding these environmental pathways that are highlighted at the bottom that we know less about. So we use wastewater sometimes to irrigate crops. We land apply biosolids, we land apply manure. These things could influence the water environment and result in exposure from recreational use. That water can be used to irrigate food crops. And of course, it can also end up in our meat products. A key aspect of our research is that we really seek to go beyond just looking at antibiotic-resistant bacteria, but to consider the genetic material that they carry. So we look at antibiotic resistance genes, or we call them ARGs. And from our perspective, from environmental engineering, we tend to think about contaminants and the spread of contaminants. And so in this context, you can think about an ARG as being a contaminant or a pollutant. And a big philosophical reason for this is because bacteria have the unique ability to share genes. And they can do this through conjugation. Transduction is mediated by viruses. And transformation is where bacteria can pick up DNA from bacteria that have been killed. So this has really important implications for us in environmental engineering as we look at water treatment processes or waste management processes that ideally a conservative approach would be to go beyond killing the pathogens or the target organisms, but to also destroy or inactivate their DNA. And here is the diagram that we've been using for our USDA project. And so we call this the Farm to Fork project. And so what we're trying to do is consider all the sources and pathways that antibiotic use in livestock might ultimately influence human exposure through the food chain, and specifically looking at produce. So here we had radish and lettuce as our model vegetables, because there's been some work on meat, but a lot less on vegetables. And we were curious if the kind of organic fertilizers that are used, so for example, manure or composted manure, if that influences the carriage of antibiotic resistance markers. And similarly, does the soil type matter? And we also took it a step further and collaborated with food scientists. And I won't talk a lot about this today, but we looked at post-harvest practices and how you wash and store the vegetables as well. And we've done a spectrum of analyses. So we look at antibiotics, we look at targets of antibiotic resistant bacteria. So we've looked at E. coli and other organisms, but we primarily have been focusing on molecular analyses. So these are analyses that focus specifically on the DNA, and that's because we're most interested in tracking these ARGs as contaminants from farm to fork. And so this involves taking the various samples that we've generated from this project. So it might be soil, it might be the surface of the vegetable that's been washed, harvesting those bacteria, extracting the DNA, and then using next generation DNA sequencing to generate metagenomes. And then I'll talk a bit about some of the analyses we've been developing to look at metagenomes. And then quantitative PCR or qPCR is nice when you have specific gene targets of interest. So the SUL1 gene that encodes resistance to sulfonamides, the INTI1, which is a class 1 integron that tends to be associated with multi-antibiotic resistance in mobile forms. We found that those are good targets for quantification. I just wanted to give a little mention here about microbiomes. So we've seen microbiomes that they're really important in human medicine. So we've realized that, in fact, we have more bacterial cells in our bodies than human cells, which is kind of humbling. And same for the various livestock we might work with, and same for vegetables as well. And so we're basically trying to figure out what shapes the vegetable resistome. So here's an overview of part of our project and gives an example of the integration. And that's really a unique aspect of what we've done, because we're all professors here at Virginia Tech with various expertise, and so we can do the animal studies. So we've dosed perlomycin and cefepime to dairy cattle, and then to beef cattle we included chlortetracycline, sulfamethazine, and tylosin in their feed. And then we did studies and collected their manure. And then we did other studies and took part of that manure and composted it. And then compared applying composted manure versus raw manure to different kinds of soil in these microcosms. And so this is a lot of work to do this, just to make sure that's clear. But it can give you an integrated perspective, and you can start to zoom in on that farm to fork continuum and get an idea of, where do we need to be focusing our attention? Is it on whether or not we should be dosing antibiotics, which antibiotics, or is it how we manage the manure? And so these are some data from our composting experiments, and in the upper left corner you can see some of our compost tumblers. And this is just showing that we had turned compost, static compost. Either way, we got nice degradation of both chlortetracycline, tylosin. Initially went up and then down, and that up part is because it illustrates sometimes you break up the parent compound, but it can be reverted back to the parent. But cefepime and perlomycin especially are highly biodegradable. Now we look at some of our metagenomic data, and I'll walk you through this. So on the right-hand side, you see a list of various classes of antimicrobial resistance. And so this is where we shotgun sequenced our DNA, compared to the CARD database to see what we found. And if you total up those genes, so working from left to right, you see the beef no antibiotic condition, beef with antibiotics, dairy without antibiotics, and dairy with antibiotics. So day 0, when we mixed that manure with the compost, all of these conditions had the highest numbers of total ARGs. However, when we composted, so we composted for 42 days, and whether it was static or turned, we saw a substantial reduction in total ARGs. So this is an encouraging indicator that whether or not you use antibiotics, that composting can help reduce the numbers. It's interesting that even with our no antibiotic conditions, though, the raw manure contained substantial antibiotic resistance genes. And I think part of that is just we've used them for so long that we've used antibiotics for a long period, and they are pretty endemic in the gut microbiome. Now here are 2 of our marker genes. So we see that SUL1 and INTI1 unfortunately actually increased during compost. So while the metagenomic data suggested benefits of composting, if we look at these genes, we might think, well, maybe the job isn't quite done yet. And then we see that if we look at the TETW gene, that one goes down. So it's just telling us that there's various targets we can look at to assess the effects of these treatments on antimicrobial resistance, and depending on which measure you pick, you might form different conclusions. And so here's a little bit about our work on trying to think about which are our ideal indicator genes. And so part of why we like SUL1 is sulfonamides have actually been the class of antibiotic that have been on the market the longest. And so they're readily detectable, and it's, maybe we don't use sulfonamides as much as a critical drug. They're still used in some cases, but they can kind of be a sentinel that gives you an idea of how antimicrobial resistance is behaving in general. And also SUL1 tends to be part of the INT-I1 operon. So INT-I1 is a class 1 integron, very important carrier of multi-drug resistance in clinical settings. And basically it has this cassette where it might have multiple ARGs. And so that's being looked at as a molecular indicator of anthropogenic sources of antibiotic resistance of clinical importance. And so you might think about this similar to fecal indicators, right? So you look at E. coli counts as a measure of overall water quality. Okay, so here's some microbiome data. So we were just curious what happened to the bacteria inhabiting the manure and compost as the compost proceeded. So from day 0 in green on the left, going right in time, we see that there's a convergence where initially there's some diversity in the microbiome of these different kinds of manures, from beef versus dairy without antibiotics. During the thermophilic phase of compost, so composting, if you're familiar, first it heats up and then it cools down. So there's especially diversity during the thermophilic phase, probably as the microbes are being shocked by that high temperature. And then eventually you get what looks like a normalized compost microbiome. So this is likely a big driver of the patterns of ARGs that we're seeing as well. And so whatever compost is doing, it is resulting in a more similar product at the end. Now, let's apply these amendments to soil. So we looked at manure at the top, highlighted in red text, versus compost to the soil. So if we look at compost at the bottom, you've already reduced antibiotics in the manure. And so you start by adding a lot less antibiotics to the soil. So we assume that's probably a good thing as far as avoiding exerting selection pressure in the environment. Now, if we look at the top at the manure condition, the antibiotics are still initially there around day 0. And so with time, if you add raw manure to soil, those antibiotics do go down. But we're concerned about what kinds of evolutionary forces we're putting at play by putting these antibiotics into the soil with the manure. And here's some metagenomic data. So we see that on day 1, when we added on the far right, that's antibiotic containing dairy manure, that it stands out compared to when we add dairy manure without antibiotics, just to the left, and the composts. And so this shows us that not only are we adding more antibiotics to the soil when it's raw manure, but we're also adding more antibiotic resistance genes. And it's here we see that the control manure added to the soil, there's less ARGs. So this is an indicator that if you know that you're administering antibiotics to the livestock, that maybe it's warranted to have some segregation of that manure and separate treatment. Now, good news at the bottom is that after 120 days, there's no significant difference in the number of total ARGs in these soil microcosms. And we picked 120 days because that's the USDA organic guidelines for harvest. So the idea is if you're harvesting crops at this stage and if they're picking up soil microorganisms, they'd be likely to carry less markers of antibiotic resistance. Now, if I go back to the previous slide, you might notice where I've circled in red and then below, the numbers went down, but the signature is different, the colors are different. So there are different ARGs, and you can see that on this next plot, which is a non-metric multidimensional scaling analysis, or NMDS. It's a way of visualizing in 2D the different kinds of ARGs present, or we call that a resistome. And so if we look on the far right here, there's a bifurcation of the yellow and the red. And what that is, it's the day 1 condition, and it's manure from dairy cows with or without antibiotics. So you can definitely see the difference in the manure in this analysis. But again, after 120 days, there's less of a difference, but the yellow and the red you can still see are slightly separated. And here's if we use our quantitative PCR analysis, looking at that SOL1 indicator gene. And I draw your attention to the far right. So this is soil where we didn't add anything, so no manure or compost. And notice how low those numbers are. So that's a sign that this is a good indicator, that it's low in the background. And then in the middle, that's the condition that's jumping up. Well, that's our raw manure containing antibiotics, had lots of SOL1 genes. And then very interesting, these different colors, the blue, red, and the green, are different soil types. And so that's a unique finding in this project, is that the soil type too can mediate the effects. So now, you know, we only looked at 3 representatives of these kinds of soil. Anybody who works with soil, there's literally a billion different kinds. But what this shows is that the soil type does matter. And in this case, the silty loam created more of a buffer to the effects, whereas maybe the sandy loam had a bit of a synergistic effect and really was associated with the ARGs lingering. Okay, let's talk about actually growing some vegetables. So Giselle Gouron, who's a postdoc that worked with us, got in the greenhouse and grew radishes and lettuce. And this was a lot of work. You got to have a green thumb. Not all of us do. And we wanted to look at all these same amendments. So this table here in blue, we wanted to look at dairy and beef and with and without antibiotics and whether they were composted. And she even looked at both static and turned compost. And she looked at just fertilizer compared to compost and manure. And then once the vegetables reached maturity, it's a bit of an art on how to get the bacteria off the surfaces and extract DNA from those, because if you just pulverize a lettuce leaf, for example, they're full of chloroplasts. And unfortunately, chloroplasts have bacterial markers, and it'll basically confound your metagenomic analysis. So just sell very gently rinsed and massaged your vegetables to extract just bacterial DNA to the extent possible. And here are her metagenomic results. So again, we see on the far right various classes of antibiotic resistance. And she's comparing the two soils, loamy sand on the left and yellow, silty clay loam on the right and green. And then we've got radishes on the bottom, and we saw they tended to carry more ARGs than lettuce, which is shown on the top. We also saw that soil effects were significant for both vegetables, especially radish, and that makes sense because it has more direct contact with soil. Now at this stage, we're getting fairly far along in the farm to fork continuum, so we're at the vegetable stage now, and here we're not seeing as much difference between compost and manure. And we see some occasional, statistically significant, but is it really significant? We're not sure, but some cases where we could tell if antibiotics were administered or not. So this is really interesting. I don't know of anybody else who's shown, the effects of antibiotic use in livestock all the way to what a vegetable microbiome looks like. And then this is the same kind of NMDS analysis that we just saw for the soil. So looking at the vegetable resistome. And so we see a few things here. So the fertilizer control I've circled here in blue. Those were pretty distinct from all the other data points, which were conditions that received manure or compost. And then we also saw a very strong soil effect. So the solid symbols are silty clay loam, the open symbols are loamy sand, and so that dividing line shows that things tended to bifurcate that way. And then here's a case where we saw an effect of whether antibiotic had been administered to the livestock. So DA is dairy manure with antibiotics, so up at the top, circled in green, the red open diamonds, and then down at the bottom, circled in green, dairy control without antibiotics. So notice those are very different. Those are both open symbols, so those are both the same kind of soil. So very interesting to see effects of antibiotics on the radish resistome. And here's looking at our indicator genes. So again, just like we saw in the microcosms, they tended to be higher in the sandy loam condition that's shown at the top. And we also saw that int I1, the red bars, in manure, int I1s on the left tend to be highest in the manure condition, the red bars. And then again, on the far right of each of these four plots, you see very low or non-detection in our fertilizer control vegetables. So again, a good indicator that these are good indicators. We've been playing around a bit with assembly. So that's where you take all the DNA sequences that you've generated. So these are metagenomes. So that means, you've got thousands of different species of bacteria, and you break them apart, take all their DNA, blast sequence it, and then basically try to put Humpty Dumpty back together again and figure out which genes are in which host organism or on which plasmid. So that takes some bioinformatics skill, but it's very interesting, and it can be useful to providing context and figure out which ARG is on which plasmid or potentially in which host. I wanted to show a little bit of lettuce results from the greenhouse and just show that INTI1 and CEL1 were similar. They behaved similarly to what we saw in the radish, especially CEL1 gene was a good indicator. Now moving into the field, and this is where things got really fun. So imagine you've got a 3-year project, you've got to get it right. it's hard enough growing things in the greenhouse, but we wanted to do field scale and get some validation. And I must say we were truly blessed with an amazing growing season, as well as an amazing team of students and technical support that put together the field plots, and the rain came. It was alternating rain and sunshine that summer. And so you can see we had a very nice harvest of both lettuce and radish. And so we have a few publications out on the field work. So on the far left, we've looked at antibiotic resistant enteric organisms in the field, and so the reference is there if you're interested. We've also looked at the runoff, so we configured these plots so that you could collect the runoff water and look at these indicators. And so definitely when the manure was fresh applied, you could see these indicators in the runoff. And then I'll talk a little bit about the vegetables that were harvested, and that paper was actually just accepted yesterday in Frontiers in Sustainable Food Systems, so you can check that out, open access. And well, let me just say it's a very similar story to what we saw in the greenhouse. So again, you see that dairy manure with antibiotics condition is influencing the microbiota of the lettuce. In this case, we're looking at the SUL1 gene. And so here we see a benefit of composting. And, one thing that we're hoping to achieve here, we used fertilizer as a control, chemical fertilizer, but, that has other environmental problems, right, in terms of eutrophication and algal blooms. And so, composting perhaps has other benefits, that you're making use of the manure product, and it's less prone to nutrient runoff. So a few thoughts to share here about some next steps and what we're working on. And so we do have a new USDA project, and this is called AgroSeek, and we have a website, and we're starting to build and populate this tool. And so this is going to focus on a computational approach to looking at metagenomic data. And so I don't know if any of you work with environmental metagenomes, but if you do, it would be great for you to email me and check this out. So the more of the community that uploads their data here, the more we can learn, and we can look at similar and parallel systems and try to identify critical control points using a computational approach. And so just some thoughts here. So I really think what we've seen so far really illustrates the importance of a systems approach. So there are things that we were able to observe because, we happen to have the right combination of expertise at Virginia Tech. We're a big land grant school, and so we could use the same manure and compost and carry it through multiple phases of the research. And it brings our attention to where to monitor. Not everybody's going to be able to do a big comprehensive project like we did. Ideally, we can figure out what are the so-called hotspots or critical control points. So, should we be focusing on restricting or modifying antibiotic use, or should we be focusing on better manure management, or do we need to think about our soil type, all of those kinds of things, because the reality is we have limited resources and we want more bang for our buck. So where should we focus? Now, a big issue here, and you probably got a flavor for it as I went along, is we don't really know exactly what we should be monitoring. There aren't exactly standard approaches for monitoring antibiotic resistance, especially if you're interested in a holistic perspective. Like, of course, we could focus in on just E. coli and just one antibiotic. So, for example, the World Health Organization is doing that, is advocating that approach as a start. So looking at ESBL, E. coli. But that's just one organism, and especially from an environmental perspective and where microbes are sharing genes, ideally we need to think long-term into looking at something more integrated. And so I have the cartoon here of the classic blind folks and the elephant, right? So each tool is giving you a different perspective on that elephant, right? So if we just look at E. coli, we might think the elephant's a tree. And if we just do qPCR of Sol1, we might think the elephant's a spear. And so I like the metagenomic approach in that it gives a little bit more holistic perspective. And ultimately, we really need to be able to inform human health risk models and have an answer for folks about, what should we worry about here. I want to talk a little bit about why we have been looking at the Sol1 genes. We saw nice patterns in the USDA project in tracking resistance from farm to fork. This idea actually originally came from our work in watersheds. And so if anybody's from the Colorado area, you might recognize this as the South Platte and the Poudre River. And they come together in Greeley, Colorado, which is a major meat packing center. And you see a lot of livestock operations, the yellow triangles, and wastewater treatment plants in red. And so we've studied this watershed for several years, and we're interested in, if you map out these sources in the watershed, and especially the Poudre River up here at the top, it has a pristine source in the Rocky Mountains. So kind of getting a sense of that gradient of anthropogenic influence. And so on the next slide, we see on the left that we did multivariate linear regression and saw a near perfect correlation for the Sol1 gene and the upstream animal feeding operations and wastewater treatment plants. So we thought that was a remarkably strong correlation for a complex environmental system. And we continue to find that the Sol1 gene, if you have to pick one gene, Sol1 and then INTI1, they're physically linked. If you paid attention earlier in my presentation, that they're both really good indicator genes. Okay, so let's say you're doing metagenomes. You want to know about all the genes there. One gene's not enough. We have issues with the public databases that, it takes time and effort to curate them and just to compare your data to the database and decide what is an ARG or not. So at the top, we see there's this so-called stringent approach, like, okay, I'm going to have really strict criteria. And so the problem with that is that you're going to end up with false negatives, right? So here's a little joke for you. This woman's obviously pregnant, but false negative would be a problem, right? You could have looser cutoffs, but you're going to have false positives, like telling this nice gentleman here that he's pregnant when that's obviously not possible. So to deal with this, we've developed a new tool, and this taps into deep learning. So we call this Deep ARG, and Gustavo is a PhD student here that was really instrumental in developing this. And this is publicly available. And the advantage of the deep learning approach is that you can eliminate false positives and false negatives, or at least get a lot fewer. And you can also discover new genes because the system learns, as the name implies. And so in our test study, we actually compared to this paper here shown at the bottom. Joachim Larsson's group had discovered 76 new metallo-beta-lactamase genes. Well, how did they do this? They used functional metagenomics. And anybody who's done that in the lab, you're cloning genes into E. coli one by one, screening for resistance, and then sequence, see if it's a new gene. They put a lot of time and effort into this. And this is an excellent paper. And we know that metallo-beta-lactamases are a big concern because they encode resistance to broad spectrum antibiotics. So the idea here was discover those ARGs before they're a problem in the clinic. Well, with deep ARG, we were able to identify the majority, about 90% of those 76 genes. And it was within minutes. So a lot of possibility with deep ARG, and we're getting a lot of users. And then I want to mention another tool called Metacompare. And so the idea here is actually built on a prior work by Jose Martinez's group in Spain. So he just put out a concept paper saying like, well, which ARGs are we really interested in? It's the ones that are mobile, right? So if you look at the data, is there evidence that it's on a plasmid or a transposon or integron? And then also, is it associated with a human pathogen? And so, like I was talking about with the assembly of metagenomic data, the putting Humpty Dumpty back together again, that you can see those neighboring genes. And so with Metacompare, we assemble the data, we look at neighboring genes, and then we assign what we're calling a resistome risk score. And that allows you then to compare different samples. And so we're working now to apply this across our library of metagenomes from the Farm to Fork project. And this can help us identify potential hotspots and where we need to pay attention. So here's an example of the validation of Metacompare. Here we compared publicly available data. Those authors called it farm effluent. I would call that a lagoon water. Hospital effluent, sewage basically, and wastewater treatment plant effluent in blue. So if you look on the right, panel B, it's quite interesting. So it was the hospital sewage that had the highest ranking of the resistome risk. And so we think this tool is quite promising, and we recently came out with a publicly available version you're welcome to check out. And here's where we begun applying Metacompare to determine the resistome risk of our farm to fork samples. So going back to those soil microcosms, where we added the manure and the compost to the soil and incubated, we see on day 1 at the top, the red, that's our raw dairy manure with antibiotics, that had the highest resistome risk scores than the other conditions. You could tell whether or not antibiotics had been administered, and you could tell whether the manure had been composted. And then far left in green is our unamended soil. So those results make sense and are in line with our expectation. And again, on day 120, we saw the resistome risk was not significantly different from the background. So that's encouraging. And like I said, we're trying to put these tools together and develop what we're calling the AgroSeek Pipeline. And we're hoping to make this broadly applicable, not just to agricultural systems, but also to wastewater and other water systems. So please stay tuned about that. And in conclusion, I think this is an urgent enough problem. We need to consider the big picture and all the options. And here we see some critical control points that we could measure effects all the way at the vegetable level. So we can consider the reduced antibiotic use, using different antibiotics, segregating manure containing antibiotics, composting. The soil type is a new dimension that might matter, what kind of soil you have, and waiting 120 days or longer before harvest. And just remembering that we all have a stake in this game. And as Gail mentioned at the beginning, this takes us back to the One Health concept. And so I hope today that I've given some good thoughts and perspective on the environmental health aspect of the One Health paradigm. So I thank you for your time and attention, and I'd be glad to have any questions that you may have for me.
Thank you, Amy. We do have a couple of questions. I encourage people to send your questions via the chat box, but I've got a couple of questions already. There's a specific question on the methodology with the vegetables. For the radishes, did you look at just the red root part of the radish, or did you look at both the greens and the radish itself when you did the analyses?
That's a very good question. So we did just focus on the taproot, the red portion that I think is more commonly consumed. I can tell you an honest answer that originally wanted to look at broccoli too. And so we found out that broccoli is a lot harder to grow than radishes or lettuce. And so they didn't crown. And so we do have some samples stored of broccoli greens somewhere. And if we have time or resources, we might look at that at some point.
Okay, and then the next question was, have you worked at all with fungal isolates, or have you been working primarily with bacterial isolates as far as resistance genes? No,
honestly, we haven't looked at fungi at all. That could be a very interesting dimension with, I know fungicides are also important in agriculture.
Thank you. And then another question is the resistome risk score that you were talking about at the end. Is that a standardized yet, or is it that score, comparison score standardized?
That's a very good question. No, it's definitely not standardized. We just published that paper about a year ago, and so it's just putting out the idea that here's a metric. And so right now there aren't any standardized approaches for monitoring antibiotic resistance, especially, as I was saying, a holistic metric that's not just looking at one organism. And so our hope is to start, well, like I said, we made the tool publicly available. People can use it. We can see what the patterns are, and then we're working to improve the method and validate it.
Okay, thank you. And then I got a question on sort of a bigger question we sort of got to at the end of how are you planning to work with folks to estimate the human health risk of the antibiotic resistance genes?
Right, that's really the million-dollar question. So I think here we demonstrated patterns, right, effects. So we can measure an effect that, you know, there is a difference in the resistome of these vegetables depending on how it's fertilized. And getting at human health risk, though, so coming at it from the other direction, you know, existing models, there's various, but they tend to focus on individual microorganisms. So quantitative microbial risk assessment is a really powerful tool, but you have to pick an organism that you're interested in. So we recently partnered with Carrie Hamilton at Arizona State University, who's an expert in quantitative microbial risk assessment. And we're planning to look at Pseudomonas aeruginosa because that's an organism that's really adaptive. It causes multi-drug resistant hospital infections, but it's also an organism that's found in soil and definitely found on vegetables. And so that's a really interesting model to start with, and there's good data. Now, there's some other approaches that involve more computational perspectives, such as Bayesian approaches that we're also hoping to look at. And then within our group at Virginia Tech, we're doing the comparative risk approach. And so I guess right now we're hoping to go at it from multiple angles. We just got a new grant from the Water Research Foundation. It's Project 4813, if you're interested and want to write that down. So Carrie Hamilton is the principal investigator on that. And I'm going to be helping lead some workshops to get input from the water industry on, what we need to do to make these risk models happen and be applicable to the water industry. So May 13, if you happen to be at the University of North Carolina, we're having an all-day symposium on antimicrobial resistance and One Health. And we're going to culminate in a session on developing these risk models. And if North Carolina is not exotic enough for you, we're going to do another one in Hong Kong on June 13th at the Environmental Dimension of Antibiotic Resistance workshop. So I really think moving this front of understanding human health risk assessment of environmental sources of antibiotic resistance is very critical for informing policy, informing what we should be monitoring and taking some concrete action.
Okay, thank you. And I'm going to sort of put a couple of questions together here, building on what you were just talking about. Sort of the problem of either dumping antibiotic waste from hospitals or from manufacture of antibiotics into sewer systems is a problem. That sludge functions basically a bioreactor. How do you reduce the problem? What do you see as being ways to deal with those two issues?
Right, that's a really good question, and I can tell you there's been some really good work on waste from the pharmaceutical industry. So Joachim Larsson's group in Sweden, the same group that looked at those metallo-beta-lactamases, they did a real groundbreaking study in India where it was a real expose showing that the pharmaceutical plants near Hyderabad were dumping antibiotics into the neighboring river at just horrifyingly high levels of cocktails and lo and behold, crazy levels of antibiotic resistance genes there. So we don't want that to happen, right? And so that brought a lot of attention to the issue, and hopefully pharmaceutical companies are being more conscientious. And it is an issue how to treat the waste. So if they're doing any sort of biological treatment, they have to think about doing it in a way that limits the evolution of resistant strains and creates a barrier to their release to the environment. And so I think this needs to be looked at more. And likewise in hospitals, there's a higher concentration of antibiotics in hospital sewage. And then, of course, the patients are ill and more likely to be carrying pathogens. There's not a lot of work on that right now, but I'm starting to see papers in the literature. In Germany, they have a major initiative. The website's all in German, but it's called HYREKA, HYREKA, if you want to Google that and if you speak German. But I've been to some of the presentations, and it's pretty compelling that you can track resistant organisms all the way from hospital sewage and then passing through wastewater treatment and being measurable in rivers and streams. So I think it's something that we should look at. I can tell you that here in the US, the Centers for Disease Control, they just had a broad agency announcement that they want to fund some research looking at that. And so hopefully we'll see some research getting funded in that area and start seeing what we can do.
Thank you. Another question that has more to do with sort of going back to the methodology. So in the methodology, when you were looking at composted manure, what was the bedding type used, or were the different types of bedding used for the animals?
Oh, that is a good question. So to my knowledge, and I'm not the vet on this project, but it's straw, and I can't tell you anything more specific than that. And I can tell you that then the compost, we mix the manure with a mixture of hay and alfalfa, and you have to get the carbon, nitrogen, and moisture ratio just right.
Okay, and then a question is, do we have any idea if there are plant resistant effects that are incorporated in the plant tissue early on that's still present at the 120 days at harvest, or at 120 days, even though they may still not be high in the soil? Does that make sense to you?
Right. I try to go back a couple slides here. Let's see. So this is lettuce from the field study. Now this wasn't 120 days because they grew fast and we just had to harvest when they were ready. But in this case, we could see that manure fertilized lettuce, and this was manure from antibiotic treated cows, carried more cell1 genes at harvest. We've tried to measure antibiotics in the tissues of the lettuce and radish, and levels are very low or non-detect. I'm not sure if I totally answered that question.
Yeah, I'm not sure if the person who asked it wants to chat and let us know whether that answered the question or not. I'll look for that. So, and then there were some more questions about somebody asked if you would repeat the information about the North Carolina conference that you talked about.
Sure. It's going to be May 13 at the University of North Carolina. So if you Google UNC Water Microbiology Conference, it should pull up. And the chair of the conference is Professor Mark Sobsey, Lobsey, and he's been instrumental actually in representing the U.S. to the World Health Organization and moving forward looking at ESBL E. coli as a environmental antimicrobial resistance monitoring target.
Okay, thank you. And I think we were going to try to get this on the CIDRAP website, up on the CIDRAP website. Plus, I think it was just that website was sent to all the participants. There's another question here on your deep ARGs. Do you have a curated database of the antibiotic resistant genomes for the deep ARG? And then the follow-up question is, which means nothing to me, but probably does to you, says, are you using CARD or ResFinder?
Excellent questions. So yes, we're actually using both. We're using all of the currently available antibiotic resistance gene databases, and then Deep ARG uses those to train the machine learning model. And then we are complementing that with hand annotation, which is an incredible amount of work. And that's why we see some of these databases sit stagnant, kind of go defunct. The folks that run CARD at McMaster University have really done well keeping that up to date, but even for them, it's a monumental effort. And so we have an approach we call ARG Minor, and this is a crowdsource-based curating approach that we're trying, and that's up on bioRxiv if you want to read about that, and you can even participate in our curating effort, and hopefully that will be published soon, but it is online.
Okay, we have, I don't know if we have, it's now sort of the top of the hour, so I'm not sure we have time for one more question. I guess there's one more question here.
Go ahead, Gail.
Okay.
One more question. Yeah, sounds good.
And it was, what would you say is the major resistance gene in dairy manure when you?
Oh, that's a good question. I mean, the SOL1 gene is one that shows the patterns, but I'd have to go back to the metagenomic data and look for myself, honestly. Let's see. So the day 0, now the manure here is mixed with the compost ingredients, so it's not a perfect representation, but we see that the tetracycline is kind of the electric blue one, and then MLS is this kind of periwinkle green color. And so tetracycline is not surprising, as it is widely used in cattle. Interestingly, we saw it in both beef and dairy, even though in this study we only used it in beef. And MLS is concerning because that's the macrolide, lincosamide, streptogrammin resistance. Macrolides are very important human antibiotics, and the perlomycin that we administered was a lincosamide. So if you look at dairy with antibiotics on the far right, notice that it did have more of this MLS resistance. So that can be a sign that it's stimulating clinically important forms of resistance. And then in mauve is multi-drug. That's another one that we see a lot of.
Well, thank you. Thank you, Dr. Pruden, for your presentation. Just a significant body of work yet to, more to be continued on here. I think there's so many questions that come up, and how then you apply this to practice and farming and, animal husbandry and human health. There's a lot of big questions out there. So I really appreciate your time today informing us of this body of work and your collaboration, and Dr. Hansen for bringing this topic to the CIDRAP ASP community. I appreciate both of your time so much, and I'd like to thank our participants for joining us and to remind them that this has been recorded and will also be posted to the CIDRAP ASP website for further viewing or sharing with your colleagues. Thank you both so much.
Thank you.
Thank you. Bye, everybody.